All right, so we're back. Let's talk about the anatomy and physiology of the vestibular sensation. This is going to be, for lack of a better term, your sense of balance. This is the situation in issues that go on in the inner ear. So our sense of equilibrium depends on input from the following sources: our visual system, the proprioceptors in the muscles and joints, and the vestibular system. The vestibular system is going to be the vestibule of the inner ear. The vestibular system includes, as I said, the vestibule and the semicircular canals found in the inner ear. These areas provide information about head position and movement, and collectively this is known as vestibular sensation. You have two types of equilibrium. You have static equilibrium, which helps us maintain our balance when the head is tilted, but the head and body are not moving. So standing still, leaning your head to the left, leaning your head to the right would be an example of static equilibrium. Dynamic equilibrium helps us maintain balance when the head or body is undergoing some rotational or angular motion, such as if you're sitting in a chair and spinning around, or linear acceleration, such as when you're riding in a car. So static, head is tilted, but the body's not moving. Dynamic, your body is moving some angular or rotational way. The utricle and the saccule are segments of the membranous labyrinth found in the vestibule. They both contain a structure called a macula. That macula is the area that has the receptor cells for head position and movement. These cells tell us, is our head upright? Is it tilted to the left, to the right, to the front, to the back? And also, is our body moving one way or another? Each of these hairs displays stereocilia and one true cilium called the kinocilium. The kinocilium is taller than the stereocilia, and keep in mind that those you'll see an image here in a moment, but the stereocilia are sort of a stair step. You've got a really short one, and then one slightly longer, then one slightly longer, then one slightly longer. And then when you get to the kinocilium is noticeably longer than the one that is right next to it. But both of these cilia types are embedded in a gelatinous mass called the otolytic membrane, which is suspended in a fluid called endolymph. The otoliths are calcium carbonate crystals found within that membrane, and they are there to increase the density of the membrane. These different hair cells will release the neurotransmitter glutamate continuously onto the dendrites of bipolar neurons that form the vestibular division of cranial nerve 8. When these stereocilia bend, that alters the release of the neurotransmitter, which alters the activity of the vestibular neurons. When the stereocilia in the utricle are vertically oriented, means up and down, they are vertically oriented when the head is upright and stationary. They bend in response to tilting the head side to side or front to back. So those stereocilia will bend either in the, and actually they bend in the opposite direction of the way you're tilting. So if you tilt your head to the right, those stereocilia will move to the left and vice versa. The stereocilia in the sacculae or sacculae are horizontally oriented when the head is upright and stationary, and they bend in response to up and down movement of the head. So remember, vertical, up and down, horizontal, left to right. That is the positioning of the hairs. The vertical hairs move in response of movement from front to back, horizontal, up and down. And there is the picture I was telling you about earlier. Here are the semicircular ducts, sometimes called the semicircular canals. There's the utricle. There is the sacculae. There's the maculas showing them. And then here are the stereocilia and the kinocilium. And you see, as you look, you got a nice little stair step pattern going here until you get to the kinocilium, and then it is clearly much longer than the one next to it. And these things will bend as a unit as we tilt our head forward and backwards, or left to right. When the head tilts, gravity pulls on the otolytic membrane, bending the stereocilia. When the stereocilia bend toward the kinocilium, the hair cells depolarize and release more glutamate, which increases the number of action potentials produced in the axons of the vestibular nerve. And when the stereocilia bend away from the kinocilium, the hair cells hyperpolarize and release less glutamate, which reduces the number of action potentials. Now let me clarify what I mean by moving toward. Now remember, all of these things are going to move in the same direction as a unit. So if they are moving that way, as you're looking at your screen to the left, they are moving toward the kinocilium. If they are moving the other direction, moving toward the shorter stereocilium, then they are moving away from the kinocilium. I don't want you to get the false idea that moving away means they're separating in some way. No, they move as a unit. They're just moving in the direction of the kinocilium or away from where the kinocilium is. And again, here is that picture showing the movement of the hairs as you move. Tilt your head to the left. Tilt your head to the right, and tilt your head to the left. The maculae are paired so that tilting your head in one direction polarizes the cells in one ear and hyperpolarizes the cell in the other ear, which allows the brain to determine which direction the head is tilting. This will be very important in a little while when we start talking about things like dizziness and motion sickness. The utricle and the saculae are also involved in detecting linear acceleration. Linear acceleration is a form of dynamic equilibrium. The maculae are sensitive only to changes in the rate of movement, not the movement itself. So they don't necessarily tell us that we're moving forward or backwards. They tell us how fast or how quickly we are moving forward and backwards. And is that increasing or decreasing? Okay, so how does inertia and the movement in the otolithic membrane and endolymph react? So you're sitting in a car in the passenger seat. You have a cup of coffee without a lid. Why are you riding in a car with a cup of coffee without a lid? But you are. The light turns green. The driver stomps on the gas pedal. You are now wearing coffee all over your shirt. Why are you wearing coffee on your shirt? The answer is inertia. The car, your body, and the cup all move together as one unit. However, the coffee, which is a fluid, is going to move independently. The coffee is also going to have inertia, but it's not going to move immediately with the cup. It's going to lag behind the cup and ends up all over your shirt. The coffee, in this analysis, is equivalent to the endolymph and the otolytic membrane in the vestibule. The movement lags behind that of the hairs in the wall of the utricle and the saculae. Those would be analogous to the cup. That bends the stereocilia, and depending on direction, either increases or decreases the neuron activity, and the brain interprets that as the body accelerating. Now, let's flip it. You're now driving at constant speed with your same cup of coffee without a lid on it. Didn't you learn your lesson the first time? Put a lid on your coffee. Where's the coffee now when the driver slams on the brake? Is it on your shirt? No, it's in the floor or in the dashboard or up in the heating vents, all over the windshield. It moves forward. This is also explained by inertia. While everyone's traveling at constant speeds, and then you slam, the coffee catches up with the cup. All right. When, however, the car, your body, and the cup suddenly stop, inertia causes the coffee to keep moving forward and out of the cup. So when the hair cells and the rest of the body stop, the endolymph and otolytic membrane continue to move forward. Those hairs then bend in the direction opposite to that when the car accelerated, and the activity in the vestibular neurons changes accordingly. In other words, if nerve activity increased when you first started moving, it will now decrease. The brain interprets this as slowing down, or the scientific term, decelerating. In the semicircular ducts, you have hairs in the anterior, posterior, and lateral semicircular ducts of the membranous labyrinth. These detect changes in dynamic equilibrium, including both angular and rotational movements of the head. The semicircular ducts are oriented to detect rotation in all three body planes. They all contain endolymph, and each duct is connected to an auricle at an enlarged region called the ampullae, or the ampulla. Ampulla, singular, ampullae, plural. And again, here are those semicircular ducts. And if you actually see these, you can see that they're sort of positioned when they're in the body in the three different planes. You have an up and down, a left to right, a front to back. And that's, each area is controlling one set of directions. Here's the support cells that hold everything in place. Here's the crista ampullaris. There's the cupula, which we'll talk about in a minute. There's the kinocilium and the stereocilia. There's the hair cell. And here are the fibers of the vestibular nerve coming into the semicircular ducts. And again, as the fluid moves, these cupulas will bend one direction or the other in response to the movement of the head. Each ampulla contains a structure called the crista ampullaris, which is a cluster of hair cells. They all have a stereocilia and a solitary kinocilium embedded again in that gelatinous mass. Here it's called the cupula. It is similar to the maculas found in the other portion of, found within the cochlea. When the head is turned, the endolymph lags behind and pushes on the cupula. The stereocilia bend in one direction or the other, either increasing or decreasing the glutamate release from the hair cell. Change in glutamate release causes an appropriate change in activity of the neurons of the vestibular nerve, and our brain then senses that as a change in nerve activity at the beginning or as the beginning of the head rotation. When the head stops, the movement of infolymph will continue to bend the cupula in the direction opposite of the direction the head was moving. So if your head was moving to the right, then those the cupulas are being bent to the left. Opposing movements has the opposite effect on glutamate release and activity of vestibular cells. Those will then signal the head has stopped moving. And again, here is the rotation. See, we're rotating this way, but the cupula is bending that way. And then it will eventually come back the other way. This is signifying to the brain that the head is moving. This signifies the brain that the head has stopped moving. And then when the head returns to its central front-facing location, the cupula will return to its undisturbed or unbent fashion. Motion sickness. Nausea, dizziness, sometimes vomiting that occurs during movement, such as riding in a car, riding in a boat, being on a roller coaster. What happens here is the brain receives mismatched sensory information. The eyes are telling the brain one thing, the vestibular system is telling the brain something else. And the body's reaction to that is nausea, dizziness, and sometimes vomiting. When you're sitting on a boat and the boat is rocking in the water, the vestibular system reports that you are moving. The visual system, however, indicates to you that you are not moving. And so, because the boat's not moving, the boat is sitting still, you're at anchor. So the boat's not moving forward or backwards. It may be listing to the left or right. The brain cannot integrate contradictory information, which causes the symptoms of motion sickness. And even in 2021, we do not understand the pathways well of motion sickness. There are some treatments for people with scopolamine patches, certain antihistamine. There's an over-the-counter drug called Dramamine, which you can get. Dramamine may be an antihistamine, I believe. But those can help with relieving nausea. You may also try sitting in the front seat of the car. If you're on a boat, look at the horizon rather than the trees and all right next to you, or the waves. By doing these things, it reduces conflicting information about the movement reported by visual systems. I was doing this lecture this morning to my live class, and told them about my sister, my baby sister, up until she was in her teens. Every time we got ready to put her in a car, we had to give her Dramamine, because if we didn't dose her with Dramamine before we drove 5 miles, we'd have to pull over and let her throw up, because she was just horrendously car sick. The pathway of the vestibular system: the brain receives and analyzes stimuli from the vestibule of the inner ear and coordinates appropriate responses. The responses have to be grouped in several categories. Number one, muscle movement to produce the changes in posture. Do you need to sit up, stand up, lay down? Cognitive awareness of head position and or movement. Is your head being still? Is it moving? Is it tilted to the left? Is it tilted to the right? Is it twisted? And then there are compensatory eye movements for some types of head movements, such as when you turn your head to the right, your eyes tend to follow that movement and go to the right as well. Most people don't turn naturally. If you're looking at an object in front of you and you turn to the right, you're not still looking at that object in front of you unless you are manually trying to continue to look. The vestibular signals then reach the central nervous system to produce appropriate responses. The vestibular signals travel to the vestibular nuclei, which are located at the pons medulla junction in the base of the brain. Those signals must then simultaneously travel to the thalamus and then to the inferior parietal lobe. Conscious awareness, this is where we get our conscious awareness of head position and movement. The cranial nerve nuclei associated with intrinsic eye muscles are also involved. That coordinates the eye movement in response to head movement. And the cerebellum and spinal cord, which coordinate muscle movement that maintains balance in response to head movement. So here's the signal again. The vestibulocochlear nerve, nerve 8, cranial nerve 8. Recall, at this point, it diverges into two nerves. You have the cochlear nerve, responsible primarily for hearing. Then you have the vestibular nerve, which attaches to the vestibule, which is, again, primarily for our equilibrium. Signal goes up the vestibular nerve to the pons medulla junction. Some signals will go to the cerebellum, again, for muscle production. Some will go to the abducens nerve, the trochlear nerve, and the oculomotor nerve. There's a nerve that has to do with the eyes. Then parts of the signal will then go on to the thalamus, where the signal is interpreted, and then go on to portions of the cerebral cortex for other different activities. All right, let's stop right there. Let me let my throat rest, and then we will wrap this chapter up.